Hello guys, welcome to my channel MBBS Studies. This is my second video on biochemistry. From now on, I will be uploading videos on biochemistry, anatomy, and physiology of second year MBBS. Now, for biochemistry, the chapter is cell. There is molecular and functional organization of cell. What is the passive transport mechanism? Passive transport is divided into simple diffusion, that is and facilitated diffusion, osmosis, and filtration. Simple diffusion is down the concentration gradient where energy is not required and no need of carrier proteins. And facilitated diffusion, here carrier mediated transport is required. Here the diffusion takes place down the gradient and no energy is required. In osmosis there is movement of solvent, and in filtration it moves from the area of high pressure to low pressure. Simple diffusion has two mechanisms, that is, lipid soluble compounds transfer directly, example urea and oxygen, and ion channels can be transported through voltage gated and ligand gated. Voltage gated are based on charge and ligand gated are based on opening and closing based chemicals, that is acetylcholine and glutamate. And facilitated diffusion, the facilitated diffusion is a type of passive transport that moves molecules or ions across a cell membrane down the concentration gradient. Examples are absorption of fructose from small intestine and transport of glucose from skeletal muscle and adipose tissue. Now osmosis, it is the movement of water slash solvent towards higher solute concentration. Clinical application is edema, saline infusion, mannitol, that is in treatment of cerebral edema, and osmotic fragility test in hereditary spherocytosis. Now, the second topic is mitochondria. Mitochondria is referred to as the powerhouse of the cell because of their role in ATP synthesis. Is a semi-autonomous organelle having its own DNA and ribosomes. Mitochondria do not exist in erythrocytes. Mitochondria are densely packed in the spermatozoa's tail. Functions of mitochondria. Mitochondria are the cell's powerhouse, storing chemical energy in the form of ATP that is released during food oxidation. The inner mitochondrial membranes, cytochrome P450 system, plays a role in steroidogenesis. Mitochondria are involved in initiation of apoptosis. The mitochondria's metabolic duties are electron chain transport, TCA cycle, beta oxidation of fatty acids, then generation of ketone bodies, urea cycle, heme synthesis, and pyrimidine synthesis, and gluconeogenesis. Mitochondria plays a role in cytoplasmic inheritance. In association with Golgi bodies, it plays role in vitellogenesis, that is yolk synthesis. Clinical application is inheritable mitochondrial DNA mutations are exclusively transmitted from mother to offsprings. Examples include mitochondrial myopathy. This is a symmetric diagram of mitochondria where this is the outer membrane, this is the inner membrane space, this is the outer membrane, this is the intermembrane space, this is the matrix, and this is the cristae. Between the cristae, that is the outer layer of matrix is the cristae, and peroxisomes and lysosomes. Peroxisomes, the structure, they are single membrane structures enclosing a granular matrix. Functions: peroxisomes contain catalase and peroxidase enzymes which destroys peroxides and free radicals, thus protecting the cells from their toxic effect. Peroxisomes are involved in the synthesis of plasmalogens, glycolipids, and bile acids. Peroxisomes are involved in the oxidation of very long chain fatty acids. Clinically, Zwellinger syndrome is caused due to an inherited absence of functional peroxisomes in all tissues, resulting in defective lipid metabolism, mainly accumulation of very long chain fatty acids and phytanic acids. Feature: cerebrohepatorenal impairment, where the child dies in the first year of life. Next is lysosomes. Structure: lysosomes are small vesicles surrounded by a single membrane. The pH inside the lysosome is lower, that is acidic pH 4.5 to 5.0, than that of cytosol. pH is around 7.4. Cathepsins are the marker enzymes for lysosomes. Functions: intercellular digestion. Lysosomes are responsible for degradation and recycling of intracellular biomolecules. Phagocytosis: they play a role in phagocytosis by fusing with phagosomes to form phagolysosomes. Digestion of extracellular materials: lysosomes also fuse with endocytic vesicles and digest the substances brought into the cell by endocytosis. Then autophagy: they are involved in autophagy, a process where they fuse with vesicles containing damaged organelles or unwanted cellular components, facilitating their degradation and recycling. And lastly, the lysosomal storage diseases. These are a group of diseases that occur due to the absence of specific lysosomes enzymes, resulting in abnormal storage of related compounds in the lysosomes. Example: Pompey's disease. It is a type 2 glycogenosis diseases, glycogen storage diseases, or you can say. Um, Now, many more topics are there relating to the cell chapter. I will be uploading it most probably to the next 2 3 videos, and please subscribe to my channel for more such updates and more such contents. Thank you.